Today we move on to chapter 19 in our Tortora microbiology text. We'll be talking about disorders of the immune system. A couple of beautiful micrograph of a couple of lymphocytes there. Um, we'll start out talking about hypersensitivities when the immune system is doing a little more than we'd like, different types of allergies. The first three are antibody mediated. The third one is a T cell mediated hypersensitivity. Type 1 is very, very quick. Anaphylaxis is called. We've already talked about that before. We'll run through again what that's all about. Type 2 and type 3 come on within hours. And type 4 is called delayed type hypersensitivity, typically, and it takes 2 or 3 days to really get rolling oftentimes. If you have a type 4 reaction that starts up in 1 day, you're going to have a major league reaction by the time it gets to its peak in 3 days or so. All right, so let's talk first about anaphylaxis. Type 1 hypersensitivities, when you have an active immune response and the type of immunoglobulin class that's produced is IgEs, they ultimately remain attached, as we said, to mast cells and basophils, and subsequent exposure to the same antigen results in degranulation of the mast cell. That's called anaphylaxis. So that's exactly what we're talking about. We've just given it a title now, type 1 hypersensitivities, a type of allergy. This can become a systemic reaction. If you have antigen that's introduced into the body systemically, say into the blood, or you eat something that goes in the digestive tract into the blood that you're allergic to, that can cause very serious anaphylaxis. Because remember, inflammation, which is what we're going to produce, causes vasodilation. And if you haven't taken physiology yet, vasodilation systemically causes a drop in blood pressure. And systemic anaphylaxis can cause a significant enough drop in blood pressure to be fatal. It can also cause bronchoconstriction. That can make it very hard to breathe. In fact, you can die from suffocation in that bronchoconstriction response. Localized anaphylaxis can be in the lungs, and you could have asthma, or you could have a more severe example in the lungs. If you inspire, say, some smoke, and you're very, very allergic to the smoke, that could cause very serious bronchoconstriction. Hay fever, people get itchy, watery, and red eyes, and sometimes hard to breathe from inhaling pollen in the summertime or in the spring. Hives, urticaria. Preventing anaphylactic reactions. Well, one thing you can do is a modern approach is to try desensitizing people to certain antigens by just giving them tiny amounts by injection and then slowly incrementing the amount that's given over a series of injections. And eventually, amazingly, it will reduce their hypersensitivity to the antigen. So that's a very promising approach. A newer idea of cause production of immunoglobulin Gs. I guess that's the mechanism is thought to by which these desensitizing injections work, is to provide IgGs that will block the production of IgEs and the setup for anaphylaxis. Type 2 reactions. Those involve complement activation and cell lysis by immunoglobulins. So let's take a look at the possible scenario here. What if you get a blood transfusion that's an inappropriate blood type? Let's say someone has type B blood, that's shown in the left-hand column. Blood types are blood groups. And people with type B blood have antibodies in their circulation against a blood cell antigen A. Right? This is what type B blood people's blood cells look like. Their red blood cells have what's called antigen B on it. They don't have any antigen A. They have antibodies against antigen A just circulating in their body, and it doesn't make any difference. But excuse me, if we transfuse them, let's say they're needing a surgery or they've had a blood loss, transfuse them with either type A blood that has antigen A on the surface, or AB blood that has antigen A on the surface, that's going to react with the antigen or with the antibodies in the circulation of our type B person, and that's going to trigger complement activation and cell lysis. So that's a type 2 reaction. It's called a cellular cell-mediated hypersensitivity. We're gonna pop those cells, lyse all those cells. And there's a number of combinations that can occur that would cause that by way of mismatched transfusions. Here's another example. Occasionally, rarely, but occasionally, when a certain drug is given to someone, it sticks to the surface of their platelets. Some proteins on the surface of their platelets, they're clotting cells or cell fragments, and then that together produces the drug acts as a hapten, produces immunological reactions. Sorry, stumbling. And so now we have antibodies against the platelets themselves with that drug on there. Antibodies activate complement when they bind to the platelets, and now we have platelet lysis. And the problem is now we're going to run out of platelets. Thrombocytopenia. That's what the title says. Drug-induced thrombocytopenic purpura. That means small bleeds. So we're going to have bleeding under the skin because there's not appropriate coagulation. So thrombocytopenia. That penia means not enough in the blood. Thrombocytes are platelets, right? They're the ones that cause coagulation, activation of thrombin, so thrombocytopenia. Type 3 are called immune complex reactions, type 3 hypersensitivities, and it's when there's a preponderance of antigen and antibodies constantly coming into contact with each other chronically, and that causes some tissue damage. And the immune complexes tend to get stuck in the basement membrane of capillaries, endothelial cells. You see lining this small blood vessel, and the antibodies are getting jammed in there like pine needles in the wintertime after vacuuming up your Christmas tree, get caught in your vacuum cleaner, can clog the hose. Well, it's a similar kind of thing. They jam in there, and when they jam in there, you have antibodies and antigens together because you have this constant presence of antigens, and that activates complement, and that attracts neutrophils. Neutrophils come in and start releasing all kinds of chemicals that are destructive and damaging, and it actually damages the microvasculature of the tissue and damages the tissue. So some examples are systemic lupus erythematosus, which is an autoimmune disease in which there are a whole bunch of different self-antigens that are being attacked by the immune system, and antibodies are constantly being produced. So there's always this presence of antibody-antigen complexes. Rheumatoid arthritis is a chronic inflammatory autoimmune disease, which there's antibodies against the aspects of the joints of the body. And then farmer's lung, occupational exposure to molds from hay can cause a chronic presence of antigens coming into the lungs, and destruction of the lungs can actually be fatal over the long term if it's not managed and treated. Finally, type 4 hypersensitivities. Delayed type hypersensitivities, and this is a T-cell mediated problem in which we bring in an allergen, in this case a hapten, which is urushiol, oil from a poison ivy plant. There's tiny little hairs in the plant that allow the oil to get onto your skin when you touch it, and then that combines with some proteins in the skin and forms an immune antigen effect. And so then we have antibodies that are, or not antibodies are produced, I'm sorry, we activate some T cells, and the T cells get all fired up, and we're going to have memory cells left in the body, so the next time you get exposed, those memory T cells are going to get all fired up and attract in some macrophages. They're going to secrete some cytokines, some macrophages are going to come in and cause tissue damage, dermatitis, inflammation of the skin. That's going to happen on secondary contact. So it's a little bit like a type 1 hypersensitivity, only instead of making antibodies, we're going to activate a population of T cells and produce memory T cells, which will get revved up the next time the urushiol oil shows up on the scene. And it can be very, very sensitive. Again, it takes days of time. They put 1 to 2 days. I would say 2 to 3 days is more typical for just bumping into a little bit of poison ivy. Most people that are sensitive to poison ivy are aware of it, and they know what the plant looks like, and so they don't usually get massive exposures to it, but it could happen. And believe me, if you get a big exposure, you will have this reaction in one day, and it's going to get worse from there. There's a picture of someone with quite an uncomfortable. This is extraordinarily itchy. It just looks kind of creepy looking when you see it like that, but it's so incredibly itchy. It's just miserable. Here's a picture of a so-called drug rash. Sometimes Delayed type hypersensitivity is against certain drugs, antibiotics. Not unusual for them to cause about a week later after treatment, cause rashes to appear on the body. And this is called a macropapular rash. It's kind of flat um, um, areas that are reddened. It's not itchy, and it's thought to be essentially the same kind of reaction, only it doesn't cause the kind of tissue damage and the kind of itchiness, but it's a delayed type reaction to certain drugs. It's just often called a drug rash, and it usually resolves on its own. So that's what this little question is about. A patient developed a rash 7 days after taking penicillin. What was this the person's first exposure? Probably no, because this is I said delayed type hypersensitivity. I think this is actually considered to be a type 1 hypersensitivity. I don't think the drug rash is 100% understood. Anyway, point is there's a hypersensitivity that produces a rash after taking especially antibiotics, but some other types of drugs, psychoactive drugs and so forth. And so probably it takes in either case, type 1 or type 4, it takes a sensitization step first, and then subsequent exposures cause the reaction. Morbilliform rashes is what that's called, a type of delayed type hypersensitivity reaction. I just stuck that in there. I looked it up quite extensively today, and it looks like there's not 100% agreement on what is the mechanism of drug rash, but maybe I'm wrong. It does involve a sensitization step first, though, which is what this question is asking about. Autoimmune diseases. In autoimmune diseases, it's either B cells producing antibodies or T cells that are creating a lot of cytokines and trouble are attacking some part of your own body. So obviously not so good. The idea is thought that perhaps during maturation, quote unquote, of the B cells and T cells in the red bone marrow and in the thymus, it hasn't been perfectly completed, so that there's some cells whose unique receptors have a slight tendency to recognize something of self, and something we didn't really emphasize, but once you start up an immune response, there's a series of steps to improve the immune response in which there's different B and T cells are selected that are even more focused on the antigen of the day, so we might get eventually quite a strong reaction against a self antigen, and then we have an ongoing autoimmune disease. Loss of self-tolerance. Cytotoxic antibodies react with cell surface antigens. We talked about that before in the context of the immune system protecting us against larval worms, and all those antibodies bind to the surface and cells that activates complement, and macrophages come in and eosinophils and start releasing toxic substances and help kill those worms. Well, that can happen to your own body in the same way. Immune complex. We talked about systemic lupus erythematosus already. If you have constant production of antibodies against antigens that are in your own body, you have this chronic presence of antigen-antibody complexes, and they get lodged in the microcirculation of, say, the kidneys or the lungs, something like that, and cause irreversible damage to the tissues. Cell-mediated. Psoriasis is a cell-mediated. It's relatively recently been described. Psoriasis is an autoimmune disease in which T cells are causing the inflammation, the itchy inflammation of the skin, thickened plaques, where the skin has just have this constant inflammation, so it just gets permanently thickened and red and quite uncomfortable. HLA. When talking about transplants and about cell compatibility, we use the term major histocompatibility complex. That's like a subset of HLA. So HLA is a term that will just be used more typically in the context of matching donors and recipients for transplants and so forth. That's what this HLA is there. So histocompatibility antigens. So we know all about those from our discussion of the immune system. So in order to do matching, we can use antibodies against different types of HLAs in order to identify them. So we said just to be simple about things and to make things understandable, we said that everyone has different MHCs or different HLAs. They're unique to your body. You bring in something foreign, boom, it's recognized and attacked by the immune system. Well, it turns out that there are groups of HLAs or MHCs that are more similar to each other. And so that's what's meant by types of HLAs. And so if you're looking for a donor for an organ transplant, for example, you'd like to find somebody whose HLA or MHCs are very similar to the recipient. And so we have antibodies against various ones, and we can take some of the donor and the recipient cells and test them with antibodies and find out which type they have. In other words, if the test antibody binds to some of their lymphocytes when we add complement and it lyses, then we know that was a positive response. We know that that antibody did recognize the HLA, and then we know which group or which type it is. So some diseases that are associated with particular types of HLA. If you have certain types of HLA, known types, you're going to have a 5 times higher likelihood of developing multiple sclerosis, an autoimmune disease that attacks your myelin sheaths of your neurons in your central nervous system. Rheumatic fever is an inflammatory disease I think it's a superantigen response, an autoimmune response after having a streptococcus pneumoniae infection, or is it strep pyogenes? Anyway, one of those streps. Thinking. I think strep pneumoniae. I'll have to look that up again. We'll see it when we come into the chapter we're going to talk about different organ systems of the body, the diseases that plague them, and rheumatic fever we'll bring that up and clarify that some more later. Anyway, you see all these different autoimmune diseases. Graves' disease is a disease of overproduction of thyroid hormone in the thyroid gland. Addison's disease is destruction of the adrenal cortex, so you don't produce the stress hormone cortisol or the blood volume and salt regulator aldosterone. Hodgkin's disease is a cancer of the lymphatic system in the lymph nodes, forms a large hard masses of lymphocytes. Didn't know it was classified as an autoimmune disease. All right. What are some ideas the scientists have about how people wind up getting autoimmune diseases? One thing that has been identified or correlated is the hormones with various types of tumors. And it's clear that certain tumors are driven by hormones. So that's one clue about estrogen maybe driving cytotoxic T cells. Another thing about it is that it's known that, say for example, people after menopause have much less likelihood of developing autoimmune diseases, or people that have had radical hysterectomies where they have their ovaries removed have much lower incidence of certain types of autoimmune diseases. So that's what brings up this theory that, oh, perhaps it's the estrogen driving some of the advent of these diseases. Another very interesting thing, sometimes maternal cells can enter the fetus during either development or during birth, and some maternal cells may survive in the fetus, and so you have a little group of some abnormal cells that are somehow cordoned off from the baby's body, the growing person's body, that could later result in an autoimmune disease because you have these foreign cells that are extraordinarily similar to the self cells, and that may ultimately produce a mistaken attack on the self cells. Inherited, certain types, as we said, of MHCs make it more likely you'll have an autoimmune disease. Environmental factors, you could get an infection. Type 1 diabetes is thought to be an attack on the islets of Langerhans by the immune system following a viral infection in which some antigen was similar enough to an antigen in the beta cells of the islets of Langerhans that there was a crossing over effect, cross reaction, killed those cells. Rheumatoid arthritis, likewise, and hepatitis C virus might be the cause. Strep pyogenes infection also possibly. That's causing an autoimmune response against the heart. That's that rheumatic fever thing. That's a strep that confirms right there. Strep pyogenes is the one. Should remember that. Exposure of T cells to antigens that are normally hidden. So some parts of your body are essentially cordoned off, and T cells don't circulate everywhere equally in your body. Like the inside of your synovial joints, for example, is pretty isolated from all the rest of your circulation and so forth. And so perhaps a severe trauma or surgery that opens up areas that are normally kind of occluded or kept away from the immune system can open up the possibility of the immune system thinking that some of the antigens present there are foreign. Uh, Testicular injury. Cytotoxic T cells may see the sperm, which are in some sense foreign because they're genetically distinct from you. They're produced by meiosis, and there's all kinds of crossing over that happens of chromosomes and things. And anyway, some antigens might be produced that look foreign and cause infertility, destroying those sperm. Transplants. We said that transplants have foreign MHCs or HLAs, and that causes them to be attacked by the immune system. Complement fixing antibodies may be produced, which will then cause activation of neutrophils and macrophages and so forth, and damaging that tissue. Certain privileged sites in the body, interestingly, are not normally, again, surveilled by the immune system, and you can do transplants there without worrying about rejection. So the cornea, for example, you can have a corneal transplant, and there's no need for any follow-up immune suppression to keep the body from attacking the replacement organ. Certain endocrine organs, see some. I don't want you to memorize these. I just think they're interesting. That are considered privileged areas, privileged tissues that can be tampered with, introduce foreign tissue into. All right, stem cells have a lot of potential application for organ replacement organs, for attacking cancers, for all kinds of things, and so there's a lot of research going on using stem cells to try to either build new tissues or create various progeny of cells that can be used for everything from drug test screening to cancers, treating cancers. So embryonic stem cells are the best stem cells to work with regarding ability to get them to induce them to differentiate into a particular cell type you want to build with, say a cardiac muscle cells or skeletal muscle cells or a gland cell for the islets of Langerhans or something. But adult stem cells have also been found to be inducible. Inducible pluripotent stem cells are stem cells produced from adult cells that have been guided by introducing introducing certain genes, causing them to express certain genes that make them act like embryonic stem cells to a degree. So here's a little diagram of a very early blastocyst, early embryo that cells can be harvested from here, stem cells that can be then induced to differentiate into any basic cell type and used for, again, engineering tissues to replace lost tissues, such as patching up a damaged heart, any number of things, kidneys, getting some kidney function going or whatever it might be. But as I said, there's a lot of promise now, especially surrounding the controversies associated with using embryos. There's lots of embryos available because fertility clinics, when people want to use in vitro fertilization to develop a pregnancy when they're having trouble, there's a lot of embryos that are left over at the end of that. So there's lots and lots of embryos in clinics throughout the country that could be used for this kind of thing. But again, some people still object to that sort of thing. So adults, here's a normal donor, isolate some tissue from that person, either fibroblasts from the skin or some peripheral blood mononuclear cells are so-called. There's some stem cell-like cells in there that can be induced. Induced pluripotent stem cells have been treated with either a virus vector or some other vector to express genes that make them more like stem cells, and they can be induced to differentiate into various different cell types and then used for cell therapy of various types. So there's lots of promise. Sometimes you can take cells right out of someone's body and then induce them to develop into a particular tissue that can be used for in vitro screening, as we mentioned. studying the mechanisms of disease, creating disease models in vitro, meaning in the laboratory. I think the reason this map is drawn the way it is, if you have a situation where you want to produce a lot of cells to do cell therapy, it takes a very long time to do all these steps to produce a lot of cells. So you'd like to have a bunch of them done ahead of time. So you start with a donor other than the patient, produce a lot of cells ahead of time, and they can be used to treat the patient in some way or another. These are all future applications. Drug screening is just now starting to become a thing in various applications for in vitro studies, but we'd like to see some cell therapies come into the main for medical treatments by these methods. Grafts, different titles or terminologies used to describe grafts. Autografts, that's when you take the patient's own tissues out, do something with them, and then return them to their body. Maybe we modify them in some way, modify some T cells to be able to attack a cancer. That's a pretty cool thing. So that would be an autograft. Isografts are when you isolate some tissue from an identical twin, kind of a rare thing, right? Allografts are the mainstay. That's what we're usually, when people talk about grafts, they're talking about allografts. Another person whose HLAs have been shown to be similar in type so that they can be used for a graft. We just need a minor amount of immune system suppression to be able to have the person retain that foreign organ. You can do bone marrow transplants. You can actually replace the immune system in someone's body with someone else's and let it take over when the immune system fails. There's a cancer, for example, a leukemia, or a type of lymphoma. It's the last ditch resort. You may decide, well, let's just wipe out that immune system of your host and replace it with someone from someone else. But sometimes there's elements of both remaining in graft versus host disease, or even if you just do an organ transplant, there can be enough immune tissue cells left in the organ that it can then starts attacking the new host, because the introduced tissue that's foreign is looking around itself saying, oh, there's foreign stuff all around us, and then you get what's called graft versus host disease. The graft, the transplant, attacking the new host. So bone marrow transplant is by no means a slam dunk as far as success rate and so forth. Immunosuppression, when people have transplants, autologous transplants, you have to suppress the immune systems, or else we're going to have all those attacks that we talked about on that new tissue. Even so, it turns out that the average lifespan, I think, of transplanted tissues is 10 years. So you see that even though we're going to suppress the immune system a little bit to try to subdue it just enough that the organ isn't immediately rejected, but you can't utterly suppress the immune system, or you'll have susceptibility to everything that comes down the pike, every infection that comes along. So it's a balancing act. What do? You give different drugs that suppress inflammation and immune response in some way or another. Cyclosporin and tacrolimus are drugs that are used to suppress interleukin-2 secretion. We talked about that, a cytokine that's used to activate B and T cells. And if we can kind of just slow down that process a little bit. Mycophenolate, mofetil, that's a good word, isn't it? Inhibits T and B cell reproduction. Sirolimus, basiliximab, and daclizumab, those are drugs that block interleukin-2 at its receptor. So we're going to have the same pathway being attacked in different ways by different creative minds at different pharmaceutical companies, but ways of suppressing immune function. Also, most of the time people are given some steroidal anti-inflammatory drugs as a combination to try to keep that foreign tissue from being destroyed. Cancer cells typically have some cancer cell or tumor cell associated antigens, some proteins that are expressing that are either abnormal or that are normally not expressed in other cell types. And so because that makes them unique, then we can find a way to target them. And T lymphocytes, cytotoxic T cells, can be taken out of the body and exposed in the laboratory to antigen-presenting cells that have been seeded with the antigens that are on those tumor-associated antigens So now they're displaying it. Expose those to the T cells. They get activated specifically. You find the one that are specific for that antigen, and then grow up a big clone of those, and then reintroduce them back into the person's body. And the idea is to have those cells then give a great big leg up on attacking the tumor and destroying the tumor cells. So there's a little picture of a lymphocyte killing a cell. Here it's cytotoxic T cell, CTL, has already acted, released granzyme and perforated granzyme, and that cell is on its way out. And here's what happened to it in relatively short order. It just deleted itself. So immunotherapy, I was kind of jumping ahead talking about that. Vaccines used against cancer, various vaccines that will be taken up by antigen-presenting cells, presented to cytotoxic T cells, and have those cells then go on and attack the tumor. Monoclonal antibodies, in a particular case of a particular breast cancer that overexpresses a particular growth factor receptor, and the growth factor is constantly being produced. It's an autocrine effect, and you can just block those receptors with an antibody and make those cells stop growing, and then maybe use something else to help kill them, and then get rid of the tumor. So rituximab is an antibody against CD20 on B cells, and cancer B cells produce that antigen on there. So if you use this antibody, you can knock back a whole bunch of if you have a lymphoma, for example, a B cell cancer growing in the lymphatic system, you can use rituximab. It's used for other types of cancers that happen to overexpress CD20. TNF alpha and interleukin 2 and various interferons kill cancer cells. So if we can find a way to express those in the context of a tumor, help destroy those. A promising idea that we have already brought up once before is linking poisonous substances with antibodies to those tumor-specific antigens, and then that will guide those antibodies right to the tumor cells, and they'll stick onto them, and then the toxic chemical will kill them. They'll be endocytosed, and then that poison is going to be right inside the cells and kill them. Vaccines, tumor-specific antigens can be used to vaccinate someone against their own tumor antigens and help kill that. Okay, so now we're going to talk about immunodeficiencies. Some people are born with immunodeficiencies. If you think carefully about everything we've learned about the immune system and how it works, every type of receptor on every cell of the adaptive immune system, of the innate immune system, any receptor, any cytokine, anything you can think of, any mechanism that you can think of, we talked about, there exists a congenital defect in that area. Somebody who is missing a particular receptor, someone who's missing a particular cytokine expression, missing a whole particular cell subtype. And so there's all kinds of congenital defects in the immune response, and sometimes they're quite complex to ferret out what's really going on. So anyway, it's one kind of category. Acquired immunodeficiencies, largely due to certain drugs, can cause suppression of the immune system. Cancers, usually when cancers of the blood are produced, the overproduction of the cancer cells quashes the function of all the normal cells. So in leukemias, typically there's one type of abnormal white blood cell being produced. They're not functional, they're cancer cells, and then all the rest of the cells are basically crowded out, and so you don't have any immunity whatsoever, and infections. So here's some different immunodeficiencies that we could just quickly look at. You do not need to go through all of these. I do want you to know a little bit about AIDS and the acquired immunodeficiency that's virally associated in that case. Some people have a selective IgA immunodeficiency, so they don't make that class of immunoglobulins. That gives a very specific kind of a susceptibility. Common variable hypogammaglobulinemia. This is a big word for not enough antibodies. Insufficient amount of antibodies. Gamma globulins are antibodies. So anyway, all these different reticular dysgenesis, not making the immune tissues, right? The reticular tissues are the lymph nodes and the spleen and the tonsils and all those different tissues where all those white blood cells live. And if they don't develop properly, you don't have the immune tissue that you need, and you don't have good immunity. Severe combined immunodeficiencies, you probably remember that one, SCID it's called. It's the bubble boy syndrome. It's in which this particular person has no B and T cells. There's no adaptive immune response whatsoever. And there's 100% complete susceptibility to infections. You can read up on the bubble boy, it's a real thing. There was a young boy who lived, I think he lived 12 years inside of a sterilized plastic bubble in the hospital, something the size of a small room that he could move all around in and had his toys and bed and everything in there. And he finally, at some point, had to have some kind of a medical treatments where they had to take him out in order to save him. And they did the surgery, but they were unable to protect him from every single pathogen or microbe of any kind. It didn't even have to be a pathogen, opportunistic pathogens, and he died of an infection. DeGeorge system syndrome. The thymus gland doesn't work, and so you can't produce mature T cells. So missing that whole cell-mediated adaptive immune response. So you see there's a whole bunch of ones, and there are many, many more. It's a very, very interesting subject. If you're interested, you can find a review article on congenital immunodeficiencies, and you can read up on some of the cool things that have been discovered. And here's a picture of a tool that's used in a lot of types of research, a lot of cancer research, in which you want to grow different types of tumors in order to either get some tumor tissue or create a model of how it could be attacked by the immune system. This mouse has no adaptive immunity. It's called a nude mouse. It's basically an SCID mouse, has no B or T cell responses, so you can inject it with tumor cells and grow a tumor on there unabated, and then you can also inject create some engineered cells of some type to introduce or reconstitute some part of the immune system of the mouse and see how it affects the tumor. All kinds of things, or you can test anti-tumor drugs. In this case, what's showing is using this mouse as a host for Mycobacterium leprae, because we can't grow them in a normal animal model, but they will grow in this nude mouse that doesn't have any immune attack on the virus or on the bacteria, Mycobacterium leprae. All right. So that ends our chapter on abnormalities of the immune system, and we're finally going to get to the chapter that I promised you last time we were about to do, which is drugs against bacteria, fungi, and protozoa and helminths.